I just want to introduce Jonathan Karr. Dr. Jonathan Karr is currently a scientific advisor at Formic Labs and works on creating transformative solutions to central challenges to biotechnology. His focus is in developing comprehensive computational models to enable personalized and predictive medicine and rational bioengineering. Previously, he served as assistant professor at the Icahn School of Medicine at Mount Sinai, developing whole cell computational models. Please welcome Dr. Jonathan Karr.
Thank you very much, Janice, for the introduction. Thank you, Veronica, and all the other organizers, Herbert, Lucian. I'll try to go quickly so that we can still end by 11, since I know that there are other speakers still scheduled afterwards. Okay, actually, yeah, that's the first slide. Okay, so I'll go quickly, but what I'd like to do in the next 40 minutes is tell you about a platform that we have been developing, which I think might provide some of the infrastructure that Paul was describing that's needed to support new modeling ecosystems. But here we're thinking of this as, rather than creating separate ecosystems for each model format and in each type of model around each simulation tool, we want to take advantage of abstraction and encapsulation and build one ecosystem, which makes it possible for investigators to share a broad range of models, broad range of simulations, simulation tools, simulation algorithms, all through one set of infrastructure. Philosophically, we feel this is important because in systems biology, there's a diverse range of methods that we would maybe like to use, and it's, I think, unrealistic that and inefficient to try to build separate ecosystems. Plus, it leads to siloing of separate ecosystems. So we feel like a better approach is to sort of conceptually pool resources from multiple modeling frameworks, multiple model formats, and kind of coalesce that all into one ecosystem rather than multiple separate ecosystems. And again, we do that through essentially abstraction and encapsulation. So the ecosystem I want to talk about includes biosimulators, which is a registry of standardized simulation tools, runBioSimulations, which is a web application for using those standardized interfaces to simulation tools to execute simulations, and biosimulations, which provides authors a place to publish standardized simulation projects encompassing models, simulations, simulation results, data visualizations of those results, and metadata about all of the above. So as Janice mentioned, one conflict of interest to declare, I, along with Michael Antonov, are starting a company called Formic Labs, which aims to develop an operating system for life science research and development. If you like the kind of infrastructure, software engineering work that is entailed in building platforms like I'll describe today, we're actively recruiting. Feel free to reach out to me by email or visit our website, where we have a list of open positions that we're currently recruiting for. If you're interested in biosimulators and biosimulations, we also welcome participants in the ecosystem that we've built. There's a number of ways that you can reach out to us, including GitHub issues, pull requests. You can email us at info@biosimulations.org. We also have weekly meetings which we welcome new people to participate in. Okay, and last thing I think before we get started, this ecosystem has been a truly community effort. A number of individuals have been involved, including developers of a variety of simulation tools, developers of multiple modeling formats, developers of a number of ontologies, and developers of several model repositories. In particular, I want to highlight Bilal Sheikh, who has been the lead software engineer for biosimulators and biosimulations, as well as Jan Moraru, Michael Blynov, Lucian Smith, and Herbert Saru, who've led the development of biosimulators and biosimulations with myself. Okay, so our agenda. First, I'll give a brief introduction to biosimulators and biosimulations, what problems they solve, and then I'll introduce the key formats that we use to abstract the details of simulation methodology, model formats, simulation tools. Those are important to understand, particularly as an author. It's necessary to understand those formats, to understand how to package up simulation projects in a standardized way that's compatible with the ecosystem that we have been developing. Then I'll run through very quickly a brief tutorial on biosimulators, run biosimulations. Finally, at the end, if there's time, I'll mention a few other related tools which I think are useful in this ecosystem. So from our vantage point, the future of precision medicine and bioengineering both need more comprehensive and more predictive models. So we're envisioning models that, in the context of cells, these could be models that represent tens of thousands of unique genes, hundreds of thousands of unique forms of proteins, a combinatorial number of interactions among all of those molecules. So we're imagining very high dimensional models that incorporate or predict behavior by accounting for the biochemical mechanisms responsible for multiple subsystems inside biological systems across multiple scales. So we believe that integration of multiple modeling strategies or multiple modeling methods and simulation algorithms will be essential to development of such models that capture multiple subsystems across multiple scales. Then we think collaboration among teams of investigators with expertise in different subsystems and scales and the experimental data and modalities necessary to collect, to parameterize such models. But we think that that type of collaboration will be necessary as well. And so we believe that presently there's too much friction to this kind of integration for a variety of reasons. While there are some ecosystems, particularly around kinetic simulations that can be expressed in SED-ML, as Paul was highlighting in the previous talk, and there's a somewhat overlapping ecosystem for flux balance analysis models, which can be expressed in another subset of SED-ML. But beyond that, for example, for rule-based modeling, that's sort of its own, largely or substantially separate silo. Logical modeling is also to a degree a somewhat separate silo. Spatial modeling, various other types of modeling are somewhat separated silos. To make it easier to integrate models and simulation methods across these various silos, we felt that it was necessary to develop a common infrastructure that made it easier for investigators to share models and simulations of all those various types. We also felt that a key piece of missing infrastructure was a platform that made it easier for investigators to share not only models, but entire modeling studies encompassing, in addition to models, simulations, results of simulations, and arbitrary data visualizations of those results. And so that led us to develop biosimulators, runBioSimulations, and biosimulations. And their goals are to make it easier for investigators to share, discover, and understand entire modeling studies, again, encompassing models, simulations, results of those simulations, data visualizations of those simulation results, and the simulation software tools necessary to execute those simulations. We felt that we also wanted to develop a platform which made it easier for investigators to reproduce and reuse simulations. So, for example, to find simulation tools that are capable of executing a particular simulation study that an investigator reads about in an article, and then once the investigator has found an appropriate simulation tool, edit the model that a user or investigator found in an article, change parameters of the model, change parameters of the simulation, or change parameters of the simulation algorithm, rerun the simulation, and repaint figures, including potentially very complex figures that were used to report the results of a simulation of a model in a paper. And very importantly, just to emphasize this again, we felt it was essential to support a broad range of modeling methodology so that all modeling formats, all simulation algorithms, all simulation tools had access to the kind of infrastructure that has existed for around SED-ML for a while, and to a lesser degree around SEL-ML and some other formats, but has been challenging to create, particularly for more innovative modeling approaches for which there are presently smaller pools of investigators working together. So just to emphasize this point about supporting a broad range, or bringing together a broad range of modeling methodology into one centralized place, here's a subset of the simulation tools which are presently available for biosimulators, and the simulation, the modeling frameworks which those, and modeling formats which those simulation tools support. What you can quickly see from this table is that those simulation tools supports every modeling framework and format. And what that means is, depending on the modeling framework and format that you as an investigator want to work with, you would need to work with different simulation tools. And then learn how to install and use various different tools. With biosimulators, in contrast, all of these details, or many of these details, essentially get abstracted away, such that investigators end up with a single standardized interface to this broad range of simulation methodology. And that allows us to build a kind of universal ecosystem around a broad range of simulation methodology. Okay. So the toolkit that we're going to talk about, at the bottom, okay. So in the toolkit that we're talking about includes biosimulators, runBioSimulations, and biosimulations. And at its core, the way that this works is we take various different modeling frameworks and model formats and simulation algorithms, and we abstract them into what's called a Combine archive. It's essentially a zip directory with a little bit of additional metadata. And once we have abstracted the details of simulation projects into a Combine archive, those Combine archives can then be executed by standardized interfaces to simulation tools, each of which know how to interpret and execute Combine archives and produce simulation results in a standardized format, which can then be further visualized or analyzed in further standardized ways. So if we drill down a little bit further, so the Combine archives contain models and simulation experiments. Models can be described in a variety of community formats such as SED-ML, SED-ML, BioNetGen, Smolden, and others. Simulations are described using the simulation experiment description markup language SED-ML. And then depending on the type of model and the simulation algorithm involved in a Combine archive, the Combine archives can be executed by corresponding standardized interfaces to simulation tools. So for example, a Combine archive which contains a flux balance analysis, a constraint-based model described in the flux balance constraints package of SED-ML, a Combine archive that has a model like that can be executed by the interface to CBMPy or CobraPy. And similarly, a continuous kinetic model, which is described using SED-ML and packaged into a Combine archive, can be executed by a number of standardized interfaces to Amici, Copazzi, Pisces, Tellurium, Virtual Cell, and several other simulation tools. And again, each of these standardized interfaces to simulation tools can produce simulation results in a standardized format, which builds upon HDF5. And then that standardization allows or enables further downstream standardized data visualization and analysis. So here's another way to think about this ecosystem. At the top, we develop biosimulations, a platform for sharing and executing entire simulation studies. runBioSimulations provides a web application for executing those simulations. BioSimulators provides a registry of the standardized simulation tools that runBioSimulations uses. And at the bottom are community formats, the foundational abstractions that biosimulators and biosimulations utilize to support a broad range of modeling methodology in a consistent way. So biosimulations presently has over 200 simulation projects, and there's about 800 more that we're working on integrating into biosimulations to make it a centralized repository for simulation projects. It will, once those 800 are integrated, biosimulations will have models integrated or simulations integrated from 5 major model repositories. runBioSimulations supports not only short simulations, it also supports computationally expensive simulations that could take multiple weeks or produce terabytes of results. BioSimulators has over 50 simulation tools registered, approximately 20 of which have verified standardized interfaces. So to drill down on these foundational standards a little bit more, from a simulation tool perspective, simulation tools are encapsulated as Docker images and their capabilities are captured with a JSON schema. The inputs to those simulation projects are Combine archives, as we discussed previously. And the outputs, as we also discussed, are simulation results in HDF5 format and data visualizations in PDF format, as well as logs of the execution of simulations in a JSON schema. Again, those simulation projects can contain simulation experiment description markup language files, SED-ML files, which describe simulations, and those simulations can in turn describe models involving a variety of model formats, and they describe algorithms using the kinetic simulation algorithm ontology, And the parameters of those algorithms are described using the same ontology. Data visualizations can be described using the Vega format, and then several additional ontologies and formats are also used in the context of this ecosystem. Okay, so I want to transition at this point to a very brief tutorial on biosimulators and biosimulations. First, I want to illustrate how an investigator can create a create a standardized simulation project, which it would be a combine archive that contains a SED-ML file that describes a simulation of a model in one of a number of formats. Then, once you have assembled a standardized simulation project, biosimulators could be used to find an appropriate simulation tool to execute that standardized simulation project. We'll then illustrate how Docker can be used to pull and essentially to install appropriate simulation tools and execute simulations using them. Next, I'll illustrate how runBioSimulations could alternatively be used to execute simulations online, avoiding the need to install and learn how to use Docker. And finally, I'll illustrate how simulations can be published using biosimulations. So there's a variety of there's a variety of software tools that can be used to create combine archives. One tool that can be used is JWS Online. It's a web application for building models and simulations. In JWS Online, after building a model, you could come to the JWS Online website and navigate to their web form for creating simulation experiments, choose an ID and a name for a simulation experiment, and click the Create button. That will then create a simulation experiment, which can then be further edited. So next, the user can choose one or more models to add to the simulation experiment. So here, a user would select from models that they have already uploaded to JWS Online or created in JWS Online. If desired, the user can describe overrides to parameters of that model. That can be overriding kinetic parameters, rate constants, or overriding initial conditions. Then the user can next describe the simulation task that they would like to execute. And that can be a uniform time course simulation or a steady state simulation. For uniform time courses, the user can then choose the initial the time at which integration should begin, the time at which integration should end, and the number of time points that should be recorded in between the start and end times. And finally, the user can use the kinetic simulation algorithm ontology to describe the simulation algorithm that the simulation should be executed using. Next, the user can describe observables, the variables that they wish to record from the execution of the simulation, say for further data visualization or analysis. We generally recommend recording all possible observables so that any possible observable can be visualized or analyzed after simulation. JWS Online can then be used to export that simulation project to a combine archive that contains a SED-ML file that describes the simulation experiment, as well as an SBML file that describes the model that was involved in the simulation experiment. So this is a kind of a preview of what the SED-ML would look like. It's not really important to understand the syntax of SED-ML because it's possible to use software tools like JWS Online to read and write SED-ML. Okay, so once you've created a simulation project or found a simulation project that was reported in an article, or maybe a collaborator sent you a simulation project, you can then use biosimulators to find an appropriate simulation tool for executing that simulation project. Now, if you're an author and you're creating a simulation project, you would probably already know which simulation tool you intend to use. So biosimulators is really more helpful in a couple of other scenarios. One scenario is you're starting a project and there's some biology that you're thinking of modeling, and you'd like to understand what simulation tools you could potentially end up using to execute the model that you are going to create. So sort of further upstream in the model development process. Another very helpful use case, or use case in which biosimulators is helpful, is if, as an investigator, you read an article reports a modeling project, and you're not familiar with the model format involved, the simulation tool involved, biosimulators can be a helpful resource for finding an appropriate simulation tool in that scenario. A similar scenario is if you're invited to peer review an article, and again, the article reports a model, a simulation project in some format that you're not familiar with. Biosimulators can be very helpful, I think, in that context. So if we come to the homepage and then we click the simulators button at the top of in the top menu, that then takes us to a menu of all the registered simulation tools. And this is a configurable table of simulation tools. The columns that are displayed can be configured using this set of controls on the left. And then to find particular simulation tools that are useful for executing specific projects, there's a number of filters that can be used to drill down to simulation tools that support particular frameworks or particular algorithms, or that provide interfaces to particular programming languages, or which can be installed in particular operating systems, and so on. There's also full text search available here, which can be used to search over all of the metadata that we have available about simulation tools. Okay, so once we identified a potentially relevant simulation tool, we can drill down on that tool. In this case, I've drilled down on Tellurium, specifically version 2.2.0. Biosimulators then gives us a brief description of the tool, tells us who created it, provides references for more information, provides links down here on the right for more information, documentation, change logs. In addition, at the top of the right panel, biosimulators provide standardized instructions for installing, and in the more recent release of biosimulators, also instructions for executing this simulation tool in a standardized way using either Docker or using Python. There's also a link here to be able to use the simulation tool, or this version of the simulation tool, in runBioSimulations, bypassing the need to understand how to use Docker or even Python. One of the most helpful pieces of information that's collected into biosimulators is information about the simulation algorithms that each simulation tool supports. Here we're looking at Tellurium. So biosimulators tells us that Tellurium supports 5 algorithms. And for each algorithm, BioSimulators gives a brief description of the algorithm, a link to the kinetic simulation ontology for more information. BioSimulators gives us information about the modeling, essentially the type of model that this algorithm supports, the formats, model formats that are supported. One of the most helpful things that BioSimulators provides is a list of all of the possible algorithms of each, all the possible parameters of each algorithm, which can be tuned, or which can be set to tune the performance of the algorithm. BioSimulators collects the type of each parameter, the default value. If there's a recommended range of values, that's collected as well. In addition, BioSimulators collects information about the variables that can be recorded from the execution of each simulation algorithm. But for example, in the context of SED-ML, that could be the concentrations of species or amounts of species, could be the values of parameters, could be rates of reactions, could be volumes of compartments. Because not all simulation tools have the capability of recording all of those possible observables, BioSimulators captures that information and captures the syntax that can be used in conjunction with SED-ML to record those observables. Another helpful thing that BioSimulators does is BioSimulators keeps track of each individual version of each simulation tool, such that users can download particular versions of simulation tools or execute simulations using particular versions of simulation tools in runBioSimulations. To ensure that simulation tools behave consistently, each simulation or each version of each simulation tool is automatically subjected to a test suite when it's submitted to BioSimulators. And in the BioSimulators user interface, we can see the results of those tests, including the logs of their execution and where tests are skipped, which would be when tests are deemed not to be applicable to a particular simulation tool. There's the reason for that skip is recorded and displayed to the user. Okay, so once we have used BioSimulators to identify an appropriate simulation tool, that simulation tool can then be pulled to your local machine using Docker. So this can be done programmatically at the command line using the Docker program. And BioSimulators labels Docker images for simulation tools in a standardized way, such that specific versions of particular simulation tools can be downloaded using Docker in a standardized way. In this example, at the bottom here, I've illustrated how Copasi 4.29.228 could be installed into a local machine by switching 4.29.228 to another version tag. A different version of Copasi could be installed. And similarly, by switching Copasi here to vCell or to Lorium or any of the other registered simulation tools, you could install a different simulation tool rather than Copasi using the same Docker pull syntax. More detailed information about the Docker images can be browsed at the GitHub container registry. So what's really nice about, I think where the power of BioSimulators really comes from is standardized ability to execute simulation tools. So rather than needing to use different command line interfaces or other interfaces to each different tool, BioSimulators interfaces to simulation tools can be executed in a standardized way. First, you would select a particular simulation tool to execute using the same standardized image tags that we looked at before. then the way that the Docker works is typically necessary to mount the directories in which the simulation project that you'd like to execute is located, as well as the directory in which you would like the results of the execution of that simulation to be stored. And then finally, you describe the location of the simulation project that you'd like to execute, which contains all the information needed for the simulation tool to know how to execute the simulation project. And you describe the location where you would like the results of the simulation to be stored. Now, as an alternative to executing simulations locally using Docker, users can alternatively use runBioSimulations. runBioSimulations uses the same Docker images, but does so behind the scenes such that users don't need to directly install and use and learn Docker. So if we come to the runBioSimulations homepage and we click the Run a Simulations button in the top right menu, we then see a web form which we can use to upload a simulation project in Combine archive format. Once that's uploaded, then runBioSimulations automatically analyzes the structure of that Combine archive. It determines the model formats and simulation algorithms that are involved, and then recommends simulation tools which have the capabilities to execute that model, the model formats and simulation algorithms involved in the Combine archive. And the users then see a narrowed menu of applicable simulation tools to the particular Combine archive that they uploaded. And then finally, users can enter an email address in which will receive an email when the simulation completes. Users can also label a simulation so that they can recall this simulation among many simulations that are run later. So if a user enters a email address, will then get an email when the simulation completes that will inform the user whether the simulation completed successfully or failed. And it'll also contain a link where the results of the simulation can be viewed. So opening that link, that'll take us to a view for the results of that simulation. That includes a menu of data visualizations that can be drawn. In addition to basic line plots, histograms, and heat maps, runBioSimulations also enables investigators to use Vega to describe arbitrary 2D data visualizations. Here's an example where we have taken an Escher map of a metabolic network, converted that to Vega, and then painted simulation results from a flex balance analysis simulation of a metabolic model on top of an Escher diagram. And Vega allows us to do this in a standardized way such that any two-dimensional data visualization can in principle be visualized inside the runBioSimulations and painted with the results of simulations. So what's really nice about this is it allows us to make diagrams repaintable. So if an author publishes a data visualization and then another investor comes along, edits that model or simulation, reruns it using Vega, we can repaint that data visualization using simulation results from a new simulation, different from the simulations that were originally used to publish the figure. Vega also allows us to capture any arbitrary two-dimensional diagrams, so that could include complex figures that have multiple subpanels or network diagrams like I've illustrated here. runBioSimulations also captures logs of the execution of simulation for debugging purposes. So each, those logs are divided into the individual component simulations of the entire simulation study. For each simulation task within the study, runBioSimulation separately captures the algorithm that was executed, the function of the simulation tool that was used to execute the simulation, the arguments that the simulation was executed with, and the standard output and error from the execution of the task. So runBioSimulations also provides a tool for managing simulation runs. And this is where you can see the names of, so when you run a simulation, if you type in a name, those names show up here. This can be used to help find particular simulation runs once you've created a large number of runs. Similar to the table for browsing simulation tools, this can also be, this table is also configurable and filterable to enable users to quickly drill down to particular simulations. Okay, so as an author, finally, once you've run a simulation in runBioSimulations and you believe that the simulation makes useful biological predictions, that simulation project can then be published using using biosimulations. That can be done from runBioSimulations. In runBioSimulations, there's a publish button. Clicking that brings up a web form, which enables you to choose an ID for the simulation project. And then the simulation, there's runBioSimulations then provides a button which can be used to publish that simulation project. Once published, the community can then find that in biosimulations by coming to the biosimulations homepage, clicking this simulation projects button, and then the project would be in the menu of simulation projects that are visible here. And similar to biosimulators and runBioSimulations, there is also a search and filter panel, which in biosimulations is hidden by default. So clicking this button here, search and filter, opens up a similar search and filter panel. Okay, so finally, I think there are, well, actually, in the interest of time, I think I'll skip this and leave time for questions instead. But thank you very much for your time and your attention. And I'd be happy to take any questions. And like I said, if you're interested in the type of work entailed in building platforms like this, we're always interested in contributors both to biosimulators, biosimulations, and Formic Labs is also hiring as well.
Thank you for your talk, Dr. Karr. Does anyone have any questions? Okay. I actually have a question. So I understand that people can run the simulations and have their results like online. How long do those results last there, and like how many results can you store online?
So simulations that are published to biosimulations, those are published permanently. And once you publish them, individual users don't have the ability to delete them. So we're kind of conceiving of biosimulations as an archive service. So once it's published, if, for example, you copy the URL for that project and you paste it into an article, for that reason, it needs to be available permanently. runBioSimulations is different. runBioSimulations the simulation projects are private to you unless you decide to share them. And you can share them using URLs similar to sharing by URLs with Google Drive. Those are also being stored permanently, but we are not guaranteeing to users that we won't at some point delete or cull our database. As far as size, each simulation project is presently limited to 5 terabytes. So for context, that is many orders of magnitude larger than the results of any simulation in biomodels, the big database, Physiome, ModelDB. It's quite a bit larger. So there's, I think, not much danger of the typical simulation projects exceeding that capacity.
Okay, thank you.
All right, well, thank you very much for having me.
Yeah, can we get another virtual round of applause for Dr. Karr? Thank you so much for your talk. Veronica, I can move on to the next speaker.